Hey everybody, welcome back to another lecture video. This one on chapter 23. So we're going to be finishing looking at essentially glucose metabolism. We'll talk a little bit about proteins and fats as we go through here and how those break down or metabolite products can factor into what we're talking about today. We looked at glycolysis last time, which was taking a 6-carbon glucose molecule and reducing it into two 3-carbon pyruvate molecules. And this process created two ATPs. Now, we're also going to finish that sort of chain reaction and see how those 3-carbon pyruvates eventually get oxidized and breathed out as carbon dioxide. So let's dive in. So we're going to be talking about the citric acid cycle. It's also known as the Krebs cycle, in honor of the guy who did a lot of study, did a lot of work on this. It's also called the tricarboxylic acid cycle, TCA. And so we're going to in-depth sort of look at all of the reactions. I think there's 8 reactions in total, so actually less than the 10 we saw for glycolysis. So this is the pathway that happens when oxygen is going to be the ultimate electron acceptor. And so we saw that there was a couple of oxidized molecules of NAD that reduced into NADH in glycolysis. And we saw what happens to those NADHs in the presence, or I should say, in the absence of oxygen. Those need to be reoxidized to then regenerate the NAD+. And that created some, ethanol eventually, and then lactate, or lactic acid, from those pyruvates. But when oxygen is present, the NADH and the FADH will have another role. And we looked at those coenzymes at the beginning of the last chapter. Okay, citric acid cycle, named for the citrate ion that is an intermediate in this reaction. In this cycle. It's actually the first product formed once we get our pyruvate molecules attached to acetyl CoA, and so or turned into acetyl CoA, and for that we need coenzyme A. Okay, so let's get to the reactions. I think this is just in general here. We're going to take the 6-carbon, eventually the 6 carbons, the 2 pyruvates, and turn them into CO2s, breathe them out. We're going to generate some more NADHs and FADH2s, and we're actually going to make one GTP molecule. And a GTP can easily turn an ADP into an ATP, GDP. And so that is going to effectively be another ATP molecule that we get before we even get to the electron transport chain. Okay, so this is another general overview. Here you're seeing what's going to happen. Our 3-carbon pyruvates, and I think we're going to actually show how they become this acetyl CoA molecule, but we're going to have 2 carbons entering the cycle. We're going to turn it into a 6-carbon molecule that is going to slowly become a 4-carbon molecule, releasing CO2s. Now, these aren't the same CO2s that came in. There are other CO2s from previous cycles. Eventually, these 2 that just came in will also get breathed out. But they're literally not those exact same 2 carbons because they're attached to a part of the citrate molecule that isn't the reactive side. So they get left behind for this cycle, but they come back and get breathed out. Because what you'll see is citrate turns into alpha-ketoglutarate. Alpha-ketoglutarate will go into succinyl CoA. Succinyl CoA will go into oxaloacetate, and oxaloacetate will actually react with acetyl CoA to make another citrate. And so this is a cyclical process. These molecules recycle, and these are various starting points for other anabolic reactions, like making proteins usually involves starting with alpha-ketoglutarate, or making saturated or triglycerides will start with one of these molecules as well. Okay, we're going to go through the reactions like I said. Oh, here you go. You can see, so proteins and fats can both be reduced to acetyl CoA, which is going to be our starting point. Obviously, glucose can become acetyl CoA, and then proteins also have a second way of entering the citric acid cycle at that alpha-ketoglutarate stage. Okay, here's the first reaction: formation of citrate. This is catalyzed by citrate synthase, and here we're seeing yeah, so I guess we're not seeing the reaction for how we got to acetyl CoA. So if you don't remember, I hope it was at the end of the last chapter, we have a pyruvate. Well, we have two of them. A pyruvate that's going to basically be reacted with our our coenzyme A. I don't remember, pyruvate dehydrogenase possibly is the enzyme. We get acetyl CoA, which is a 2-carbon group, and we lose a CO2. And so this happens twice because there are 2 pyruvate molecules. So now we're at acetyl CoA, which has lost a carbon. It only has 2 carbons, and that's this guy right here. Okay, so we see that reaction. We form citrate. We regenerate acetyl CoA. So this could, I'm sorry, we regenerate coenzyme A. It just rolls off the tongue better, acetyl CoA. Coenzyme A gets regenerated, so it can go back to this reaction and keep doing this. Reaction 2 is an isomerization reaction. Citrate into isocitrate. Reaction 3 is a decarboxylation. This is the first CO2 that gets removed. And this is also the first NAD that gets reduced into NADH. Another decarboxylation. Alpha-ketoglutarate gets decarboxylated into succinyl CoA. Again, another NAD+, becomes an NADH. This reaction also involves another coenzyme A. And we breathe out a CO2. All right. Hydrolysis, part of reaction 5. Here, the succinyl CoA gets hydrolyzed back into coenzyme A, succinate, and this is what we would call substrate level phosphorylation. So we're going to talk about oxidative phosphorylation, which is making ATP, phosphorylation being transferring the phosphate group from ADP into ATP. And that happens in the electron transport chain, oxidative phosphorylation. This is happening in well, it's not exactly the cytosol. This is happening in inside the mitochondria, but this is happening at the substrate level. So this guy's a substrate. This is a substrate. When these substrates react with each other and create high energy molecules, we call that substrate level phosphorylation. So this is, again, going to make an ATP easily. So we count that as some of the energy produced. Reaction 6 is going to get us back to our, or start getting us back to our starting point. There's one more reaction after this. Succinate is going to reduce FAD into FADH2. This will carry 2 hydrogens. And we're going to get fumarate. Hydration of fumarate gets us malate. So there's one more after this. 8 reactions in total. And finally, the oxidation of malate into oxaloacetate. Now, oxaloacetate was what reacted with acetyl CoA in the first reaction to give us citrate. So we've gone all the way, one full circle here. Now, this whole chain, this series of reactions happens twice, because every glucose molecule had 2 pyruvates, and each pyruvate goes through this cycle. So everything we're talking about here, we'll have to double it in terms of the big picture of how much was made by one glucose. But again, another NAD becomes an NADH. So let's total up what we have. Well, here we can just see it, but we'll total it up here in a second. Where's the FA? Here we go. So we've got a couple of electron carriers to take electrons to the electron transport chain, and we made a GTP or an ATP equivalent right there. Here's a nice reaction to kind of show all of what we got back. We regenerated coenzyme A. We got 3 NADHs, an FADH, 2, and a GTP, as well as those 2 carbon dioxides that were breathed out. When you count the 3rd carbon dioxide that was breathed out, getting from pyruvate to acetyl CoA, that essentially accounts for all of the sugar's carbons. So the entire glucose was now effectively breathed out. There's a couple of regulation sites here in this reaction, similar to glycolysis. These are obviously the enzymes that are going to be regulated. Reaction 1 has it is increased in its activity, or what we call activated by ADP, and then it's also inhibited by NADH, ATP, and citrate. And that should make sense. If there's lots of citrate, we don't need to do this reaction 1. If there's lots of ATP, we don't need reaction 1. If there's lots of NADH, we don't need to do reaction 1. Reaction 3 is also a regulation point. ADP, again, an activator, NADH and ATP, a deactivator. And in reaction 4, alpha-ketoglutarate dehydrogenase is activated by ADP, but it's inhibited by NADH and also succinyl CoA. So just kind of to illustrate again, often the products themselves of the enzyme are going to be deactivators for these enzymes. It's a neat way how your body regulates the sort of speed of these reactions. Okay, pause this, give it a shot. Okay, moving on. So now that we've generated a bunch of electron carriers, let's go see what they're actually going to do. So we're going to have to total up the amounts of NADH from both glycolysis and from the citric acid cycle, as well as those FADH2s, and actually sort of see how they ultimately get more energy by going to the electron transport chain. And we're going to look at some of the complexes within the electron transport chain. So, okay, these coenzymes are going to now give their electrons to different electron acceptors, which we're going to use that energy to pump hydrogens into the intermembrane space of the mitochondria. So we'll have to look at an image here to kind of refresh our memories on what that looks like. So let's get in there. Let's see. This is what we got coming into the electron transport chain. We've got those 2 ATPs that we already formed in glycolysis, as well as these 2 NADHs. Pyruvate into acetyl CoA generated some more NADHs that I neglected to account for. Citric acid cycle generated 6 NADHs and 2 FADH2s, as well as a GTP or ATP equivalent, which we should call, what do we say here, 2 ATP equivalents with the 2 GTPs from the 2 cycles. All right. The electron transport system has a few complexes. We're going to look at each one. Let's see. This is talking a little bit about where those enzymes and complexes are located. Some coenzymes are attached to the inner membrane. Some are bound to the inner membrane. Okay, we're going to see a picture. All right, so remember, we've got the mitochondria with their outer membrane, and then their inner membrane has all these little folds in it. And this creates something called the matrix. And all of the reactions that we've been talking about, the citric acid cycle, is happening here in the matrix. We're in the matrix. Okay, the intermembrane space is all of this area in between the inner and the outer membranes, right? And so first off, there's NADH and FADH2 being generated inside here because that's where citric acid cycle happened. Glycolysis, though, happened out here. And so there's a couple NADHs that can't get in here yet, and we're going to have to explore how we get those NADHs, the one from glycolysis, inside here. But what we're talking about at the moment are these membrane-bound complexes and coenzymes that are on the inner membrane. And so what they're going to do is they're going to pump hydrogens into this intermembrane space. And this intermembrane space then is going to build up with hydrogens. We're going to use that like water in a dam to then drive this ATP synthase. So now I'm down here. So intermembrane space is right here. This is the matrix in here. And these hydrogens are going to be allowed to come back through this protein. This protein is going to spin. There's a neat video on YouTube that'll show this thing spinning. I'll try to link it in the description. We've got ATP being generated, and the hydrogens that come through here are going to join with the electrons that are coming in from this electron transport system to join with the oxygen to make water. And so that's the waste product of all of this. Now notice that there are a few complexes. There's complex 1, which basically is going to oxidize the NADHs. Complex 2, which is going to oxidize the FADHs. Now, complex 1 pumps hydrogens with the energy that it just received. Complex 2 doesn't do that. Complex 2 just takes these electrons from FADH, gives them to cytochrome Q, who is then going to get oxidized by this, there's like iron sulfur proteins and whatnot. We're going to look at that in complex 3, which is going to use that energy to pump hydrogens. Because this is not as far away from ATP synthase, it's kind of like when they call it further, it's not as far upstream, or it's further downstream, I forget how they say it. It doesn't generate as much energy as NADH. NADH gets way more energy per electron than FADH does. Anyways, we're going to have a few more electron transfers, cytochrome C back and forth between coenzyme 3, complex 3 and complex 4. Complex 4 is also going to pump electrons across, and then complex 4 is going to give these electrons to oxygen, where it's going to await essentially the ATP synthase, or complex 5, to let those hydrogens through to form water. So here's complex 1. And again, this is just summarizing essentially what I just told you happens in a little more detail. And here we're just looking at how NADH in its reduced form regenerates NAD+. So remember, we need NAD+ to be able to have glycolysis continuing. That's why in the absence of oxygen, we would rather make lactic acid than do nothing. So then you can see coenzyme Q here becomes reduced, pumping some hydrogens across the membrane. Complex 2, again, just takes the electrons from our carrier here, regenerates FAD. Coenzyme Q then is going to give these electrons to cytochrome C. To complex 3, iron-containing proteins, and then those get transferred to cytochrome C, which goes back and forth between complex 3 and 4. And this is that image that we were looking at right at the beginning of this section of this chapter. Kind of looks like the heme core at the center of our hemoglobin here. But this is the cytochrome molecule. And it actually does have an iron core, just like our hemoglobin does. So this heme group is essentially the way that this iron is kind of held here is really the same as it is in hemoglobin. Okay, so we get another oxidation. Here's that reaction. We see that iron actually gets reduced from iron 3+ to iron 2+ within this cytochrome C, and that's going to oxidize our coenzyme Q. There's the electron transfer, and we get some hydrogens pumped across. And right about now, we should be towards the end. So before we get to the end, let's talk about where the NADH that from glycolysis went. So there's a way of essentially getting those electrons transferred across, and it's called the malate aspartate shuttle. So let's take a look. So this is just reminding us there was some NADHs made by glycolysis, so we want to see where how much those will get in the electron transport chain. NADH itself cannot pass through the mitochondrial inner membrane, so there needs to be a way to get the electrons across, and so malate dehydrogenase will catalyze the reaction of oxaloacetate and NADH to make malate and NAD+. Then malate itself will cross the membrane. And then malate dehydrogenase, which is the same one that sort of did this oxaloacetate to malate reaction, will turn malate back into oxaloacetate. So it's just a reversal. And then you can see what happens here, oxaloacetate. So let's follow it. So we've got oxaloacetate here in the cytosol. We've got NADH. We want to get those electrons inside. So malate dehydrogenase creates malate, and we get NAD+. Malate crosses, and malate goes back into oxaloacetate, but the NAD pluses that were inside the matrix become NADH. So effectively, the NADH has gone up by 1 for this process. So this is how we shuttle, quote unquote, the electrons into the mitochondria. Okay. And then because so then to get to complete the circle, my bad. To complete the circle, oxaloacetate has to be turned back into aspartate, which can also cross the membrane, and aspartate can become oxaloacetate. So that's the full cycle. Sorry. All right. So let's see. So this is a little out of order. I suppose this should have been back before the rest of this, but this was the reaction that I was trying to tell you guys about. The 2-pyruvates becoming acetyl-CoAs, breathing out some CO2s. We didn't go over this when I was telling you initially, but I interjected a little later and reminded everybody that we've got 2 NADHs that actually do come from this process. So let's count up the total ATPs from the entire glucose molecule. So NADH gets you about 2.5 ATPs. So 2 NADHs, they're going to count that as 5 ATPs. Now I think this book's a little out of date. I think they've changed the number, and that happens a lot. Citric acid cycle, we got 2 ATPs effectively substrate level, 2 FADHs and 6 NADHs. So we'll have to count all that up. And you can see that essentially here. So this is just taking into account the citric acid cycle. And this is for 2 turns. So this is counting both of the pyruvates that go through this process. We get 20 ATP just for citric acid cycle. And then if we look at the big picture, glycolysis got us 7, right? Because the NADHs got us 5, and then there was already 2 ATPs we got in that process. Making the acetyl-CoA got us 2 more NADHs, and that gets another 5. And then there's the 20 that we just looked at, so altogether 32. 32 ATPs from the oxidation of a glucose molecule. So I don't know how much of the next chapter will go in detail over, but if we don't go into that much, I want to say here, we will get more ATP for oxidizing amino acids or proteins, and for fats. So those are the more high energy molecules. This is why you want to limit them in your diet, especially if you're not that active, because without anything to do with that energy, it all just gets turned into glucose, or converted into glucose through gluconeogenesis, but then it goes and gets stored as glycogen. And if you don't use all your glycogen, that gets converted into fats for long-term storage. All right, so 32 ATPs is what we get all together. Now, in terms of efficiency, let's see. 1 mole of glucose should actually generate, now in the lab using a calorimeter, if you remember back to Gen Chem, you should be able to get about 690 calories or kilocalories. But in terms of our bodies, we only get about 230 kilocalories. So we're not the most efficient. We do get a lot of energy, and because of this big change in energy just from the citric acid cycle being tacked on to glycolysis, which a lot of some tiny organisms can't even do more than glycolysis. We've been able to do a lot with that, right? There are elephants, there are blue whales, and then we've got the brain powers that we have because of all this energy that we're able to commit to our bodies and building structures and whatnot. The rest of that energy dissipates as heat, which is good, right? Helps keep us warm. But it isn't the most efficient. Of course, it is more efficient than a light bulb, or at least the older version of the light bulb. All right, 33% efficient. That's actually not bad. All right. So, if you're wondering what I mean about the light bulb, typical 100-watt light bulb generates or converts about 1 watt, which is joules per second, of energy into light, and and dissipates about 99 watts as heat. And that's the old filament light bulbs. The new LEDs and whatnot can have a higher efficiency, but yeah. So we are way more efficient than a 100-watt light bulb. That's for sure. All right, guys. I will see you in the next chapter.